Okay, let's go ahead and get started. Good to hear all the happy chatter. Missed that last year. If you're looking for a micro lecture, you're in the right place. I'm Professor Susan Leonard. So EMST 12, hopefully you're in the right place. I want to start out with a few COVID related things. Obviously we have to keep our masks on, so you're all doing so far so good there. And also I just want to remind you that you should all be self-assessing. All right, so you should have been doing that all last year as well. It's really easy if you just get a button right on your, get the app or whatever, right on your phone. All right, so you should be doing that every day that you come to campus. If you're never sure if your symptoms warrant, isolating or whatever, that will tell you. So use that as your guide. Obviously, we're more than 6 feet apart, as if that was a magic number. But in other classes, you're probably having a seating chart. There's 140 of you, and we're not going to do a seating chart. I'm not going to pass around any kind of attendance or anything like that. I am, when I remember, going to try to just take a few pictures just in terms of contact tracing. I don't know that I would be able to recognize any of you with your mask on or whatever, but I don't know. Maybe we'll try that, see how it goes. It's kind of, the whole contact tracing thing is going to be kind of up to you. So I may get notified that a student tested positive, and maybe that student will tell me, oh, I was sitting in the lower left-hand corner. So I might be able to announce in class, anybody over here, like, be especially vigilant for symptoms, or maybe you should go get tested. I'm encouraging you to sit in about the same place every time, as much as that's possible, and to kind of know who you're sitting around. All right, so if you don't know who you're sitting around, I'm going to give you a minute here to introduce yourself. So go ahead and introduce yourself to somebody next to you. Go. Okay, good. That's a good start. Okay, we'll do that periodically. It is good for you to be here in person. I am going to be recording all my lectures, and don't see that as a free pass just to stay home. I just would encourage you to come. You'll be more engaged. You'll get to know people. You'll find people to study with. I'm hoping that I get all the content when I record things, but I really just think it's best if you are here. So encourage you to do that. All right, so we're just going to hit the highlights of the syllabus. I did post the syllabus on Blackboard. I would encourage you to read the syllabus. I know there's some things that I'm just required to put on there and whatever, skip over that if you want. But I talk extensively about the homework, about ways to succeed in this class. I'll touch on that a little bit this morning, but you want to go back. That's a good reference document. Feel free to ask, questions, but again, as we go, but always consult the syllabus first, I guess. I've tried to spell things out there as best that I can. All right, my office is up on the 3rd floor of Henry. It's right down the hall from the labs. So if you're in lab, I'm assuming that maybe about 90% of you are in lab right now. So I do kind of refer to what we're doing in lab a lot, but don't worry if you're not in lab. I'm not testing you over things that you only do in lab. There is a little bit of overlap between the two courses though. So it's nice that if you are in lab right now, it can be helpful. My office hours, it just worked out best for my schedule. I had a big gap in my schedule on Mondays and Wednesdays. I have an 8 o'clock lab and a 2 o'clock lab. So I had a big gap. And so I put all my office hours then. That's not to say I am available lots of other times. Usually right after lecture, I'm available. So that's a good time. If you have questions or need something to clarify, we can go up to my office. I will be in on some Fridays. Really the only time that I'm not available is if I'm in lab. I have 3 different labs that I'm teaching, 213, or right before class. I like that time right before class, make sure I have some lunch, I'm fueled up, and I'm reviewing what's going on in class. So don't be afraid to ask. We can Zoom. That I'm pretty flexible. A little bit about me. So this is a pandemic picture from last summer. That's what I look like without a mask on. Hopefully we'll eventually get to take our masks off, who knows. My husband teaches in chemistry, so some of you may have had Dr. Leonard. You can just call me Professor Leonard. I don't actually have my PhD. I have a master's. So I answer to Dr. Leonard, but I'm not really a doctor. So Professor Leonard works. We have 3 kids. The oldest is in medical school through the Navy. So if any of you are thinking about going that route, I'd be glad to talk to you about that. She starts her rotations this winter and has to go to Hawaii for that. She said, somebody's got to do it, Mom. So it's been a good gig for her so far. Our youngest is in his last year at Michigan. He's an engineer, got his pandemic haircut there. And our middle daughter has autism. She lives at home with us and works a little, goes to school a little. So that's a little bit about me. I've taught this class enough that I'm losing track. I think this is my 7th year of teaching lecture, and I've taught lab for about 15 years. So I've been doing this a while, still have a lot to learn, still learning a lot. But I do find it extraordinarily interesting, and it's probably the most applicable class maybe that you'll have right now, right? Living in a pandemic. Hopefully this class will help you make even better decisions about things that you do every day. So we'll talk a little bit shortly about what we're going to cover this semester. I've had, last time I looked this morning, about 1/3 of you have already gotten that code and are logged into the Mastering Microbiology site. So if you haven't done that yet, you can go to this link. I think that you can purchase the code online. You can also purchase the code from the Laker store. And then you need this number to get into our class, basically. And that's in the syllabus, it's here. It is $100, and that, like everything, has gone up in the last few years, and I feel kind of bad about that. It is a good book. It's not too complicated. It's not too dumbed down. It's really a nice, I think, level for our introductory microbiology class. So we do follow along with the book pretty closely. That code will get you the e-text, which has its advantages. There's animations right in there, so it's actually not too bad to navigate through. If you really like having a physical textbook, then you can purchase that for extra. I would we're working out of the 6th edition, but the 5th edition is really close, and you could probably get a good deal on that. Even the 4th edition is just some page numbers off. So you can get an older textbook if you really like to have a textbook. This also includes the homework. So about 10% of your grade is the online homework. Most of those, I have chosen all those homework questions for you. So I have picked the ones that I think are most applicable to what we're covering in class, and not just a bunch of stuff from the textbook that you won't see on an exam. Most of the questions are to help you review, right? So the more times you're exposed to something, the greater chance it's going to get caught in your long-term memory. Some of the questions are just to kind of get you interested. There's some case study type questions. So I think it's good. And again, students like that they have a little homework component of their grade, and their grade is not just exams. So, you know, if you have a problem with a particular question that you think isn't fair or whatever, by all means, ask me about it. And we'll go from there. Okay, and just let me know if you have any technical problems getting into that, and I can try to help you troubleshoot a little bit. By the way, can everybody hear me okay? Okay, just wave your hands if you can't. The microphone, I can't even tell if it's on or not. It might be a little bit, but okay. It sure beat, I taught in the gym last semester, so this room sure beats that. How will you be graded? I hate to kind of hit that with you on the very first day. I really went back and forth on how to structure this, because last year I only did 3 exams. And in some ways you might think, oh, the fewer exams, the better. I get so stressed out. That's true, but when I have fewer exams, then each one is kind of higher pressure, and there's more to study for each one of them. So in the end, I decided that really it's better for you to have 4 exams rather than 3. I always like to weight the final exam a little extra, because that's what the experts say, that if you have a student that's doing better and better as the semester goes on, that you should reward that, right? Maybe even ideally, I would have that first exam worth a little bit less points to keep tossing that around. But so it does seem like kind of a lot of exams. I've tried to structure that so they don't fall at the same time as your lab exams. Okay. How does those homework points work? You have in the order, I think it's actually a few less than this, around 500 possible points, actual homework questions. So, and again, this will be different. There's 4 sections of this class. Other instructors will do this a little bit differently, but as long as you get 80% of those homework questions right, I'll give you the full 60 points of homework. All right, so the reason that I have done that rather than just have you, whatever homework points you earn, that's the kind of percentage that you get. The reason that I don't like to do it that way is because I found that students are spending hours and hours and hours doing their homework to try to get every little point squeezed out of that. And I don't really think that that is the best way to use your time. I like the fact that you have to kind of, most of them are multiple choice, so you have to think about it, you have to pick an answer, and that helps engage your brain a little bit. But you can guess every once in a while, and still get something out of it. So my effort was just to try to not make this so you're spending way longer doing homework than you're productive. All right, so I go a little bit more in depth in the syllabus there, about how that homework is graded and things, but the website actually grades that. And I don't actually get that number really till the end of the semester. So you can kind of judge how you're doing. If you're getting 80% or a little bit better on every homework assignment, then you're doing well. If you're not completing them, or you're getting 70%, then you need to plan to do better to get that full 60 points. Okay, so again, that's just a little cushion on your grade that may be helpful. All right, so how can you do well in this class? Well, I will post the PowerPoint outlines. I try to do that at least the night before and hopefully well before that. I always have to like kind of read through them and make sure there's nothing that I want to change, but I try to get those out early enough. You may choose to print them out. I would say maybe 2 to a page, sometimes 3 to a page. They get kind of small if there's some kind of intricate diagrams on there. You may decide, print them out and take notes on them. You may find it, you can take notes right on a laptop. I will be writing in content as we go. So what I'm going to test you over is what I already have printed on there and what I write in. All right, I try not to test students on just maybe things I say in passing. I won't be testing you over reading that I don't specifically cover in class. Okay, so you'll see as we get to the schedule that I have page numbers kind of reference for the book for everything that we cover. And the book is a great reference, especially things that we go through in class that are a little bit more complicated, like viral replication. You might want to go back and read the book's couple pages on viral replication just to kind of give you a little better picture of what's going on. But again, if it's in the book, even if it's in the reading and I don't cover it, then that's not necessarily something that you need to know for the exam. I also have learning objectives, and I've posted those already for the semester. Not a bad idea just to print those out. It's about 9 pages front and back, objectives for the whole semester. It seems like a lot, but basically some of these I've made up myself. A lot of them I've taken right from the book. So I've got the references to where they are in the book. And what I do when I make up an exam, I look at those objectives and I say, okay, I'm going to think of a question that kind of fits into that objective. So I kind of make sure I go through here, okay, well, I covered this, and this, and I try to, make the exam balanced so that I don't ask a bunch of questions about one thing and nothing about other things. So that's a good tool. In the past, some students have gone through and like written out answers to all those. And I'm not sure if that's maybe for a lot of students, that's a good thing to do, but it's definitely something that you would want to be aware of and look at and maybe realize, oh, I don't know what that's talking about. Maybe I better look at that a little bit more carefully. So that's another tool that you have. I would encourage you, those homework assignments, most of them will take between like 30 minutes and 60 minutes. At least that's what the publisher will kind of guess that. I would encourage you to do that right after we cover that material in class. So we should get through chapter 1 in class today. It's just kind of introduction, history type stuff. Tonight or tomorrow, I'd say take a look at it and see if you can get that homework done. It's not due till the day of the first exam though. So you have some latitude. I would say don't wait till the night before to do all your homework. That's when the students get in trouble and don't finish it. And it just doesn't do you as much good because you're kind of rushing through it. So you can always go back and look at it again to study, but I wouldn't put it off. In general, I would say learn the terminology. So with any biology class, you're going to have to learn a lot of terms. And when we learn those terms, then we use them. And so if I'm using a term like, say we've talked about opsonization, and then I use that term and you don't know what it is, then you're just going to be struggling to kind of catch up. So if you come across a term that you don't know what that means, look it up. That's how we become sort of fluent in thinking and talking about microbiology. It's hard to read a research paper if the abstract, you don't know a bunch of those terms that they use even, in the introduction. So hopefully we'll become fluent in this terminology. Part of the final exam is just matching terms, okay? And in your objectives, I've tried to boldface some of the terms that I think are probably going to be new. So just to draw your attention, hey, do you know what that term is referring to, what that means? So, okay. And like I said, I would certainly encourage you to read the textbook. It's a good bit of reading. You know, it might be a better use of the textbook, again, to go back and read things that are a little bit more complicated. A lot of us, I know when I read something the first time, I don't really remember a lot from it. So if I know a little bit about the topic first, and then I go back and read, it sticks with me a little bit better. Okay, and also, I came upon a book this summer. It was like something, it was called something like How to Be a Great Learner or something like that. Again, talk about it here in the syllabus. What's it called again? Oh, Learn Like a Pro, right? A brand new book. I just came upon it in the library, and it had some really good tips for college students. So the last page, I kind of wrote a little synopsis of what I learned from that book. And basically they said, if you're trying to learn something, probably just reading over your notes is absolutely the worst way to go about doing that. And that might be, that's how I used to study, I know, in college, but really you need to be as active as you can. So if you're reading the textbook, read a paragraph and then stop and think, what was that paragraph about? If you have no idea, then, well, you need to read it again, pay a little better attention, right? So maybe look at a page of your notes and, you know, oh, this topic was different kinds of microscopy. Okay, so quiz yourself. Do I know what those different kinds of microscopy are? Do I know what the cells look like? All right, you have to be constantly testing yourself to see if you know what you were just looking at. Okay, practice teaching it to somebody or your stuffed animal, or you just have to find active ways to kind of engage your brain so that you start to move that information, not just to remember it for a few hours, but more long-term memory. So take a look at those tips. There's also some tips about procrastination, which, you know, all of us do from time to time. All right. So what are we going to do this semester? I just kind of took some screenshots here of the syllabus. This course has a very clinical bent to it. So if you take a look at the American Society of Microbiology, what an intro microbiology course is supposed to cover, even though we have an enormous amount of content in this class, we don't cover everything that we are supposed to for an intro microbiology class. Because again, we kind of skew it. A lot of you want to work in health professions, so we talk a little bit more about the bacteria that cause disease, or a lot more. We don't talk very much at all about environmental microbes. So the kinds of bacteria and fungi that you might find in the soil or in the Grand River, right? We don't really get to that very much. Okay, so we start out kind of with the basics, and we skip around a lot here. You can see 1, 6, 4, 3, lots of different chapters we're skipping around. It's a little confusing, but we're trying to kind of match up with what you're doing in lab right now. Okay, so we kind of get those, you know, lab and lecture to gel up a little bit, can be helpful. And we're just kind of hitting a lot of basics here. How do we grow microbes? How do we look at them under the microscope? I mean, what are they? What are their structures? So just kind of a lot of basics here. How do they grow? How do they metabolize? How do they reproduce? So the first half of the course is kind of the basics, and as well as we'll get to viruses here in about a month, a little basic information about viruses. So that's kind of the first half, and then the second half gets a little bit more application, a little bit more, hey, how do we kill those microbes that we don't want around? So I want to first of all say that not all microbes are bad. You might get that feeling from the way we teach this course. We could not exist without microbes. You have a ton of them in your body that help you be healthy. You would not be healthy without them. They decompose things. They make all sorts of products for us. So, but unfortunately, we do concentrate a lot on how to kill them when we want to do that. All right, we talk about how to kill them on surfaces, a little bit about antibiotics, how to kill them in our bodies. And then we spend a good bit of time on our body's immune system. All right, we have a separate immunology class here at Grand Valley, but maybe a lot of you won't get to taking that. So we have a good, pretty thorough introduction of immunology in this class. And then at the end of the course, we pick out some specific infections and talk a little bit more in depth about them. So for instance, we'll talk about tuberculosis. So what does that organism look like? How do we see it under the microscope? How do we diagnose it? What kind of strategies does that organism have to grow in our body? So we get really specific then. I did want to point out this exam 4 that's your Tuesday right before Thanksgiving. All right, so I know that day, if you're in the habit of taking off early for vacations, that's kind of a bummer. I certainly didn't want to wait till after Thanksgiving to give you an exam. And again, I thought about just going to 3 exams, but I really think that having 4 gives you less material for each exam. So I just wanted to throw that out there. I am hoping to give all of our exams in person. That's a change from what we've had to do all last year. So don't just assume that I'll be willing to give you that exam if you're in the UP that day or whatever. Okay, I want you to be here. I am giving makeup exams for good reasons. Certainly talk to me. I should get an email, if you're having COVID issues, and I'll be reasonable and work with you about that. I will give makeups probably within about a week of an exam. And after that, oftentimes we'll just say, okay, we're just going to not take this exam. We're going to double the final, which is not a great option because that puts a lot of pressure on you for the final. But sometimes circumstances, I've got students in the hospital or whatever, then that's what we have to do. So that day, I just wanted to draw to your attention. We do have class a week from today. We used to get a 2-day Labor Day break, but we do have class next Tuesday, and then you get a fall break a little bit later in October. So, all right, that's sort of my introductory spiel. Any questions that you can think of that you might have about our course structure or for me? Okay, well, don't be afraid to jump in. I know it's a big class. It's kind of hard to hear with the mask and everything, but don't be afraid to jump in. All right, so chapter 1. This is just a little bit of history that we're going to touch on to be, well-informed microbiologists. We're going to think about what do we mean when we say a microbe. I'll just go ahead and define that. A microbe is something that you can't see with the naked eye. All right, so microbiologists study things that are too small to be seen with the naked eye. So that's, we talk a lot about bacteria, but that includes other kinds of organisms as well. From time to time, I like to look at kind of famous experiments and look at their structure a little bit so we can learn about scientific just experimental structure. And so there's a famous kind of experiment of proving how fermentation works with Louis Pasteur. We'll talk about some famous people that have contributed to us understanding the causation of disease and what we can do to prevent it. All right, so the people that I talk about, those are the ones that I picked out of chapter 1 that I want you to be responsible for. All right, so the first time we saw microbes, right? If microbes are things that are too small to be seen with the naked eye, then we need to have some way to magnify what we're seeing. Antony van Leeuwenhoek, he was, at least, he's the one that we give credit to for discovering or making the first microscope. Okay, so I liked this picture from the previous edition of the book here. That's what his early microscope looked like. And it looks nothing like what we would think about a microscope looking today. The lens is actually right here. Okay, and then this little pointy part here, he would put his specimen, like if he was looking at a fly or whatever, he could stick it on that kind of pointy part. And then you'd lift it up and he would look through the lens. All right, so it's pretty amazing. He got pretty good magnification with this, somewhere between low and medium power of our microscopes upstairs. So somewhere, you know, up to about 200 times magnification, but it was pretty good. He looked at a lot of things, drops of water, his own bodily fluids, like he looked at all sorts of things, submitted his work, was recognized by the Royal Society of London, and was a very recognized scientist of his time. But actually his first job, he was a cloth merchant. I just gave you the years here just so you kind of have an idea. Don't memorize years, but this was, you know, kind of mid to late 1600s, so a long time ago. And it probably seems odd, or at least it did for me. Why would a cloth merchant make a microscope? Well, he got really good at grinding these lenses. And he was born in the Netherlands. And if you think about stereotypes that you might have of Dutch people that you may know today, some of the stereotypes that I have is, oh, they're tall, maybe they're blonde, they're really neat, they're clean, they vacuum their house all the time. Another one is that they're kind of careful about their money. All right, if you go Dutch on a date, that means that, you know, the guy doesn't pay, that they each pay for their own. Well, that was probably true, the whole being careful with his money thing about Antony van Leeuwenhoek, because he wanted to make sure that the people he bought cloth from weren't cheating him. If he was paying for a good quality of cloth, he wanted to be able to see that it actually was good quality. So he made these lenses, again, got really good at it, and just looked at all sorts of things. The problem was he wouldn't teach anybody else how to make those lenses. It was kind of like his guarded secret. So we didn't progress very quickly and get better microscopes very quickly, again, because he wouldn't share that. But if you think about all the things that he looked at through his lenses, again, he wrote this all up, published this. We could group these things that he saw into about 6 categories. Okay, so if you think about what are microbes, well, he definitely saw bacteria. What else? What else would you think of as being microbes? Anybody want to? Yeah, fungus. Yes, definitely are some. Of course, there's some mushrooms and things that you can see, but there are fungi like yeast that are small. What else? What else are microbes? Viruses. I'm going to put that in parentheses because he couldn't actually see those, right? So Leeuwenhoek, we knew that there were viruses before we could actually see them. Electron microscopy wasn't developed till the 1930s, but yes, so definitely I would include that. What are the little things that if you scoop up some pond water, you can see swimming around with a microscope? Probably done that in biology class at some point. Algae, that's a good one too. What I was thinking of was the protozoa. Okay, we spend a little bit of time on protozoa, not very much. And the other thing that we will talk more about what they actually are is archaea. They're kind of, some people might think those as being the same thing as bacteria, not exactly the same thing. Leeuwenhoek also saw some, we'll say, small multicellular animals. Okay, you know, maybe tiny worms, things like that, that we could consider microbes. All right, so Carolus Linnaeus, he was a Swedish botanist that his life overlapped somewhat. He was a little bit younger than Leeuwenhoek, but he is famous. He's generally considered the father of taxonomy. Okay, so he's credited with kind of helping the scientific community organize a whole lot of data that it had about organisms. All right, and we're going to talk a little bit more about taxonomy probably Thursday and its importance. But you know, he recognized that in order to be able to identify organisms and name them in a kind of reasonable way, there had to be rules that everybody would follow. For instance, if he decided that everything needed a Swedish name, then you know the Frenchmen and the Germans might not like that. So he decided everything needs to have a Latin name. That's sort of the common language of science is Latin. So we'll talk more about that. All right, so a little bit more about some basic information about these organisms that we're going to study. Both the bacteria and archaea are considered prokaryotes. So that's a term that you should be familiar with. Not just, these are organisms that lack a membrane-bound nucleus. All right, they don't have things like endoplasmic reticulum or lysosomes. Okay, so we're going to talk really extensively about bacteria. Here's a couple examples of bacteria. This is an organism called Deinococcus, which actually lives and metabolizes in radioactive waste. So that's kind of cool. I always think it's cool. We can think about ways to get bacteria to clean up our messes or do things for us. Staphylococcus, that's what this middle picture is. That definitely can make you sick. We probably all had staph infections. Both the bacteria and the archaea are generally about 10 times smaller than a eukaryotic cell. So there is almost always a pretty good size difference there. Here's an example of an archaea. It's a methanogen. There are archaea that actually produce methane. So that's kind of a bizarre thing. A lot of the archaea in the past has been thought about, oh, these are organisms that live in very extreme places like thermal deep sea vents or polar ice caps or very salty places like the Great Salt Lake. But we now know that there are archaea that live in our body as well. So it's not just weird organisms. One thing that's so far, we think that archaea do not cause disease. Okay, we haven't found any yet that cause disease. So for that reason, we don't really concentrate on them in here. Talk about them a little bit in terms of classification and things, but there are structural differences in the cell wall and how they reproduce themselves that will distinguish them. Other organisms, some eukaryotes that we talk a little bit about, some microscopic fungi. This is a scanning electron micrograph or a really, really close up of the mold that produces penicillin for us, Penicillium chrysogenum. All right, so it naturally secretes penicillin, which of course has helped us a whole lot. This is Saccharomyces cerevisiae. That's the yeast that does a lot of alcoholic fermentations for us. So fungi can be very useful to us. Here's just some pictures of some protozoa. They are everywhere, especially in bodies of water. Some of them cause disease. Malaria, probably the best known protozoan that causes disease. And algae, we really only mentioned a few times, even though they are responsible for, being really important in food chains and producing oxygen for us. They don't cause disease for the most part, unless we get way too much of it. There can be some toxins produced by overgrowth of algae. But these are on the right here. These are diatoms. These are marine algae and some freshwater algae as well. Cool to look at through a microscope. Parasitic worms do have some of them microscopic stages, like their eggs, but we really don't have time to get into parasitic worms in this class. If you're really interested in parasitology, we do teach a parasitology course every fall. Dr. Graham teaches that, and he's had a lot of the infections that he teaches about. So it's an interesting class. I'd like to sit in on myself sometime. And then, of course, viruses. You all probably know more about viruses than what you really want to already. But of course, a few basics here. All right, so again, we'll talk much more about viruses later, but they are considered acellular. That means they don't have a cell structure. They don't have a typical cell structure. They may share a few things in common with cells, but very little. Okay, they either have RNA or DNA, never both. This was really mind-blowing when we discovered the first RNA virus. Here's something that can, make more copies of itself, and it doesn't have any DNA. All right, so in this drawing over here, this is actually a drawing of the SARS-CoV virus. So the cause of severe I'm blanking, the respiratory syndrome that came out in the early 2000s. I've used that picture for a long time. This is right here. This is the RNA of the virus. It's got kind of a protein coat around it to protect it. Some viruses have an envelope around it, and that's pretty much it. They're pretty simple. They just kind of take over living cells. Okay, they are obligate intracellular parasites. So that means they have to live inside living cells. Okay, they can't reproduce anywhere but inside a living cell. All right, so in lab, we are not growing up viruses on medium. Okay, we could grow viruses in bacteria cells. And here are some examples of some really crazy looking viruses that infect bacteria cells only. Okay, but we can't grow viruses just on a collection of nutrients. Okay, they parasitize cells. They take them over and eventually kill them. All right, so that's a little introduction into the kinds of organisms that we're going to study. I want to leap ahead here to talk a little bit about Louis Pasteur and some of his contributions. Okay, so that's a name that we'll probably mention now and then. Louis Pasteur was a French chemist that lived in like 1850 to maybe the late 1800s. And he made a lot of contributions, not just kind of understanding how fermentation works, but he contributed to what's called the germ theory. And a lot of people kind of contributed to that, but that just means that we started to understand that it was germs that cause infectious disease and not things like bad air or bad odors. That was a prevailing notion for lots and lots, many, many years. Okay, so we were just in this golden age of microbiology from about 1850 to 1915 or 1920. We started to make lots of progress in understanding what caused disease and maybe kind of what to do about it. Pasteur also made the first vaccine, the anthrax vaccine, and he also made a rabies vaccine. So a lot of contributions. But one of the first problems that he tackled was, how does fermentation work? Okay, the wine industry was very important in France at that time. It still is. And the thing was, they didn't really understand how grape juice became wine, that fermentation process. Like, they didn't know if it just happened spontaneously or how that worked. And so when something went wrong, they lost a lot of money trying to make it right again because they didn't understand how it worked. So in the wine industry, or some vintner hired Louis Pasteur to consult and try to help them understand how this fermentation process worked. Okay, so they knew that grape juice that fermented, when they took a look at that, when it became alcohol, and they looked at it under the microscope, they knew that there were microbes in there. So they kind of suspected, oh, there must be some microbe that has something to do with this. But Louis Pasteur made 4 hypotheses, and then he did some tests to try to figure out which one of those would be closest to the truth. So he said, well, maybe fermentation just happens, okay? And these microbes aren't necessary. So to test that, he took some grape juice and heated it up enough to kill everything in there, sealed it up, and then looked at it on the next day under the microscope, determined that it was still grape juice, it hadn't changed into wine, and there were no microbes. Okay, so it doesn't just happen on its own, right? It was still grape juice. So he rejected that hypothesis. Then he hypothesized, well, maybe it's just the air. Maybe oxygen is a chemical reaction and the grape juice just automatically turns to wine. Well, he designed these really cool swan-necked flasks himself, where basically you could, heat up that grape juice and make it still be exposed to the air, but anything that was floating around in the air would kind of get caught in this bend in the swan neck flask. Okay, so they tested that grape juice after a day. There's no microbes in it. Is it wine? No fermentation and no microbes. So, okay, it's not the air that ferments the grape juice. Then he said, well, maybe it's bacteria that are doing the fermenting. So he sterilized the grape juice, added bacteria in, tested it the next day, and there's lots more bacteria, but he didn't get the good tasting wine that he expected. The medium that grape juice was super acidic. All right, and this is something that you're going to test over and over again if you're in your lab, but bacteria do ferment, but when they do, it's almost always acidic end products like lactic acid. All right, so we're often testing that in the lab. It's just a simple pH test. Oh, if it's acidic, then that organism must have been fermenting that sugar. So you'll learn that if you're in lab, especially. So you had to modify that hypothesis. Well, yeah, they're making some change, but it's acidic, which is not what they wanted. What if, is it yeast that ferment the grape juice? Sterilized the grape juice, added the yeast, and then they found that was producing the wine, the good tasting wine that they wanted. It was actually bacteria that was contaminating the yeast that was making the wine kind of go off. Okay, so that process of kind of heating up the grape juice And then adding the yeast, that process of heating became known as pasteurization, saved the wine industry. We use it today, obviously, to make sure that milk is safe to drink. We pasteurize lots of other things like apple juice, ice cream, eggs sometimes. They don't actually heat grape juice anymore to make wine. They do that chemically because the heating kind of changes the flavor of the wine, apparently. But it was an important start. All right, a contemporary of Louis Pasteur was Robert Koch, and he's another name that we're going to mention every now and again. Koch was German, Pasteur was French. They fought on opposite sides during the Crimean War, and they didn't like each other. Instead, they worked on a lot of the same problems, but instead of kind of helping each other, they were adversaries, which is kind of a sad thing. But Robert Koch is best known for making advances in etiology of disease. And that just means causation. Okay, so he was best known for being a very meticulous planner in his experiments. Kind of later in his life, he kind of made some missteps and said that he'd found a cure for tuberculosis, and he hadn't. He made some mistakes, but for most of his life, very meticulous. And he discovered the first microbe that he proved actually caused a disease. Okay, so that we just kind of take that for granted now, but back then that was a big deal. Again, it was like the odors or whatever that they thought caused disease. Okay, so he named this organism Bacillus anthracis that was a causative agent of anthrax, which was a huge problem in killing lots of farm animals, especially back in his time period, again, like late 1800s. Okay. He also discovered the causative agent of tuberculosis, which up till then people thought was a cancer. It was very slow growing and slow acting and developed over many years, ran in families. So when he proved that it was actually an organism. Okay, so some of his meticulous experimental designs gave rise to what he called Koch's postulates. Okay, so your book talks about this, but it doesn't have kind of a nice little picture. So I got that from online. But he formulated these things that need to be satisfied in order to prove that some agent actually causes a disease. So the steps that he took here, he said, well, number one, that germ has to be present in every case of the disease. Okay, so you have to have an animal model. You can't be doing these tests on humans, or at least we don't do that anymore. He took some mice that were sick with some infection through, taking some kind of tissue sample or blood sample. He was able to grow those microbes up on some media, and then isolate each one of those different organisms that grew up in what we call pure culture. I'm going to define that again later. We're talking about growing organisms in the lab, but that means only one organism in one physical place, like the petri dish. Okay, so the second thing is that the germs have to be isolated and grown all by themselves. All right, so just to do those two things, there had to be a lot of advances made in culturing, in actually seeing them, being able to stain cells so that you could see them under the microscope to help us understand. And then, and I forgot to say that this germ that he's trying to isolate, it shouldn't be found in healthy individuals. So if you've got a mouse that's healthy and you take the same tissue sample, you don't find that same organism. All right, then you take that organism that you isolate it, it's all by itself, inject it back into healthy animals. And if that animal gets sick and you're able to isolate it again, the same thing, then that is proof that that germ must have caused that disease. Okay, so this was a good first step. This doesn't work, obviously, in everything that causes disease today. We know lots of diseases are not infectious. They might be genetic or caused by a toxin, for instance. Viruses have to be grown up in a very different way than this. There are some infections that are caused by lots of different organisms, like pneumonia or ear infections. So, not everything is going to satisfy Koch's postulates, but again, this is a really important first step to make people understand that it was microbes oftentimes that were making them sick. Hans Graham was in Robert Koch's lab. And like I said, they had to get better and better at seeing things under the microscope. He developed a special stain that we know as the Graham stain that enables us to see organisms really well. And you can see in this picture that some of the cells are pink and some of them are purple. Okay, that tells us some information. It gives us some information about those organisms, whether they are positive for the Graham reaction or negative for the Graham reaction. Nothing to do with the charge of the organism, but there's differences in cell wall structure that allow some organisms to hold on to some dye and others to get very quickly decolorized. All right, so if you're in lab, you're going to do a lot of Graham stains. It's just kind of the first question we ask when we're trying to identify an organism. Is it Gram positive or is it Gram negative? Okay, because we have certain antibiotics that work better for Gram positives or better for Gram negatives. Okay, thinking about preventing infection and disease. There's two big areas of advancement that we've made that help us avoid getting sick. Number one is improved hygiene. This other thing is vaccines. And you could argue which one of them have saved more lives. Both of them have been really important. And we didn't always understand that being cleaner would keep us from getting ill. There are some sort of pioneers that helped us understand that. Ignaz Semmelweis, he was a Hungarian doctor. So again, 1848, this is really before we understood that germs cause disease. Still, it wasn't, maybe there was some suspicions or whatever, but we didn't really understand that very well. He noticed that in his hospital, they had a very high rate of women that died of childbed fever, women that gave birth in the hospital. It was almost like 18% of women that got this infection and died of it. And he noticed that doctors were going straight from doing autopsies to delivering babies. And he thought maybe there's something, he called them cadaver particles, that were causing women to get sick and die. So he started advocating hand washing. And he saw the death rate just plummet down to like 2% for doctors that had washed their hands. The problem was it made doctors kind of mad that he suggested that they were the ones causing the problem. And they didn't really understand why hand washing helped. So he wasn't really accepted. He ended up dying in an insane asylum when he was 47. So it didn't end well for him, but his work was known. And about 40 years later, an English doctor, John Lister, knew about his work. This was late enough that the germ theory was a little bit better accepted. And he advocating using carbolic acid, which is phenol, in the operating rooms. Okay, so phenol at that time was sort of like the gold standard for killing microbes, or it was for many, many years. So this drastically cut down on infections when you put it on wounds in hospitals, or he would spray it in the air during operations. Surgeons would wash their hands in carbolic acid. The reason that we don't use phenol very much anymore as a disinfectant is because it's really toxic. So unfortunately, it did cut down on the infection rate, but doctors' hair got bleached. They had respiratory problems. Their fingernails fell out. So thankfully, we have other things now that help cut down on infection rates. And also Florence Nightingale, she was a nurse during the Crimean War, and she was a very, she thought that hygiene was really important. So she insisted that her wards were kept very clean, that wounds were kept clean, and she kept good statistics about the death rate of her soldiers, and was found that they lived a lot longer and had less infection in her wards. So all these things just helped us understand that hygiene is important. Okay, and in terms of vaccines, Edward Jenner, this is 1796, so we're going back pretty early here, of course, before they really understood what caused disease, but he's credited with the first vaccine, and that was to smallpox. He noticed that milkmaids that often got an infection called cowpox almost never got smallpox. So he took some cowpox scabs and he stabbed them into some kid, and then inoculated the kid like a week later with smallpox. Not quite the way we do clinical trials today, but he didn't get smallpox, and that was, still not a great vaccine because of the risk of getting infection, just the way they kind of inoculated people. But it was a whole lot better than what they did before, and take smallpox scabs and kind of scrape them into somebody else, which was likely to give you a milder infection of smallpox. Could also kill you. So anyway, we've been getting better and better at vaccines ever since then. And one more name here, Paul Ehrlich. He was also in Robert Koch's lab in Germany. He's kind of the first one to think about treating infection. That was a very difficult thing. We didn't have really any idea where to start, but he systematically tested many, many different chemicals, trying to find one that would cure syphilis. Okay, he was searching for a magic bullet. I don't know if I spelled bullet right there. That doesn't look quite right. Anyway, that's gonna bug me. Yeah, okay. A magic bullet would be one that you would shoot out of your gun that would kill your enemies, but not your allies. All right, so he was looking for some chemical compound that would kill the bad bacteria in our body, but not harm our bodies. All right, so he searched for many different compounds. He finally found one that was effective against the bacterium that causes syphilis. It had arsenic in it, so it was still pretty toxic to people, but there was some use there. Like, it did kind of weigh out a little bit in benefit of the person that had syphilis. So again, beginning steps in trying to treat disease. One more person that we have to mention, John Snow. He's considered to be the father of epidemiology. Okay, epidemiology is kind of a trendy field right now. Everybody's interested in that. Of course, that's we study like how diseases are passed. Well, it's more to it than that, but kind of how they're distributed and spread. Really, epidemiology is a lot of legwork and a lot of asking people questions and a lot of statistics to try to parse out what information is really important. There was a cholera epidemic in London, 1854. And he suspected that what was causing it was something in the water was causing it. That was what he suspected, but he couldn't really prove it. So he went around, he got a map, he put out, the water pumps are in blue here. He put those on the map, and then he went around door to door asking people, is anybody sick with cholera in your house? You know, where have they been? What have they eaten? What have they drank? You know, all kinds of questions. And then he kind of put down where the people were that were infected with cholera, and he noticed that they were clustered around the Broad Street pump, right? So they took the handle off the pump and the epidemic subsided. All right, so the power of statistics there and just asking the right questions. All right, so those are kind of the historical people that I wanted to draw your attention to. That's pretty much, there's some more interesting stuff in chapter 1, but that's kind of the highlights that I wanted to pull. Any questions, comments so far? Okay, we got a little time left. I'm gonna skip into chapter 6. Again, skipping around a little bit to try to match up with what you're doing in lab tomorrow and Thursday. This is actually the middle of chapter 6 in your book. We're going to come back to the rest of chapter 6 maybe in a couple weeks. But here we're looking at, well, why do we want to grow bacteria in the lab? And, you know, that's just so we can learn about them, right? So we can maybe help fulfill Koch's postulates if that's necessary. How do we do that? And that's what we're going to start talking about. That term pure culture, I alluded to that already. That's got some important terminology to know. And then we'll talk about some different kinds of media. We can use different kinds of media depending on what our goals are for what we're trying to grow up. All right, so just some terminology that sometimes trips up the beginning microbiology student. We can use that term culture both in an active way and as to describe something. So we can use it as a verb or a noun. If we're going to say, I want to culture something, then that just means that we want to grow it up. Okay, I'm trying to culture some E. coli. Okay, I want to encourage it to grow up. I want to make more of it. If you refer to something as a culture, like right here, this is a culture of bacteria. That just means the place where the microbes are growing. Okay, so that could be a petri dish, that could be a test tube, probably one of those things with media in it. Here's another two words that kind of sound alike. We can talk about inoculate something, again, active, or we can refer to something as an inoculum. If we're going to inoculate something, that just means we're going to introduce organisms into a culture. Okay, that means we're going to take some cells from an already growing culture and start them in a new culture. We're going to inoculate those cells into some new medium. Okay, the inoculum is those actual cells. So that's the organism. Okay, so the organism used to start the culture. And the medium is just the collection of nutrients. That's what the cells are growing on, where they're getting their nutrition. And maybe includes some kind of hardening agent or solidifying agent. Maybe we'll get into that in a minute. Okay, so our organisms are growing up on medium. Okay, in this case, the medium looks a little bit red. There's probably some blood in there. Blood is a really nutrient-rich source. So sometimes we add that to our microbiological media. Okay. Sometimes students mix up the term inoculate with incubate because they kind of sound alike. All right, but when you incubate something, that just means you're going to put that culture at its ideal growth temperature. Okay, so for most of the organisms that we use in lab, that's going to be somewhere between 35 degrees Celsius to 37 degrees Celsius. All right, if we're trying to grow organisms that typically grow in our body, then we want to incubate them at about body temperature. Okay. And different organisms have different requirements there, right? Some of them like it colder, some of them like it hotter. Okay, so in this picture right here at the top, it's a picture of somebody taking a loop in order to inoculate a culture. So when you're inoculating, you can't really see anything on here. You're really just putting a few cells on there. And of course, you can't see the cells with your naked eye. But once those cells grow up, typically they will grow in discrete packets that we call colonies. Now, that's not always the case. Sometimes cells just kind of swarm over your whole media. But when they're growing on solid media, oftentimes they grow in these discrete packets called colonies. So basically one cell here that we can't even see with our eye, when you give it enough nutrients, it divides. One cell becomes two, and each one of those cells becomes two. Okay, so they multiply so many that now you can see them with your naked eye. Okay, and if one of these colonies isn't touching any other colony, we call that an isolated colony. And we often want to get isolated colonies because those are colonies that all the cells in it are basically clones of each other. They're definitely all the same species within one colony. Okay, if you get a bunch of colonies growing together, sometimes it's hard to tell if there's several different kinds of organisms in there or just one. Okay? And we often like to work with just one kind of organism at a time. All right, so a little bit about the basics about the media that we use. Oftentimes, we're going to use liquid media in lab, and we refer to those as broths. Okay, so a lot of the media that we make up in lab, we buy it in powdered form, add the appropriate amount of water, we stir it up, and then sterilize it. Okay, and then pour it into our test tube or whatever that we want. Okay, so nutrient broth, that's a good, just kind of all-purpose media that we use a lot. Tryptic soy broth is another one. But broths are really good to use to grow up large amounts of bacteria. Large amounts of cells, we'll say. Okay, they grow quickly, they've got plenty of room to move around, they're bathed in nutrients, put them in a nice warm place, you get lots of cells. How can you tell if cells are growing in liquid media? Here's some nutrient broth where there's nothing growing in it. You can see how you can kind of see right through there. The more cells you get growing, the more turbid it gets. You can see how this one here is kind of cloudy. Okay, that's because there's bacteria growing in there. So often you can tell with your naked eye if there's something growing in there. You don't know if it's one species or if it's 20 different species, but there's something growing in there. Sometimes the growth tends to kind of settle out at the bottom. And to see that, you might have to give the test tube a good finger twack to get it all mixed in there. Okay. If we want liquid media to be solid, that often is a big advantage because, of course, on solid media, you can see the colonies And you can easily tell if you have a pure culture. So if you have only one kind of cell, right? And again, that's what we call having a pure culture. Okay. Got 1 more minute. What makes a solid media solid? We add something called agar to solidify. Okay, agar is a polysaccharide that's found or derived from the cell wall of a certain kind of seaweed. It's actually red algae. Okay, and we add that as a solidifying agent in order. Agar is something that bacteria can't metabolize. So when we add that, if the bacteria ate it, then the agar would break down and the media would liquefy. But because they can't eat it, it stays solid. And again, then we can see those discrete colonies. All right, so thanks for your attention. Have a good day. I'll hopefully see you on Thursday.